All right, everyone, we're going to go ahead and get started. So I'm happy to introduce our speaker today, Erin Evans, who will be giving the talk Endogenous Retroviruses and Where to Find Them, A Journey Through Health and Disease. Erin is a third-year PhD student at the Department of Microbiology and Immunology at UBC. She was born in the UK and grew up in New Zealand. Her work focuses on ancient viruses embedded in the human genome and their possible role in causing multiple sclerosis. Outside of the lab, she is a swim coach, plays water polo, and champions neurodiversity acceptance, showing that society has room for and thrives with all neurotypes. Erin was also one of our co-chairs of the resident member series last year, along with Nico Jimenez, and they have graciously handed the reins to Ruth and me. Speaking of which, Ruth is back from her trip to Mexico. Welcome back, Ruth, and she'll be co-leading these events with me going forward. Now, before we begin the talk, we'd like to acknowledge that we are gathered on the traditional, ancestral, and unceded territory of the Musqueam people. And with that, let's hear it for Erin.
Okay, thank you all for coming out here today. And I hope you'll learn something about endogenous retroviruses and where you might find them. So, we've all heard of the microbiome. It's the 38 trillion bacterial cells living in and on our bodies. And there's been a lot of talk in the news about how they affect your health and how they can affect your lifespan and how to take probiotics to boost your microbiome. But less people have heard about the virome, which is the 380 trillion viruses that make up your human virome. And this is a largely understudied resident population in your body. So what type of viruses make up the healthy human virome? We have bacteriophages, which are bacteria-eating viruses. We have endogenous retroviruses, which is the topic of the talk today. And anelloviruses, which are entirely harmless viruses that are a major part of the virome. And so before we get too far, let's first think about what are viruses. So here we've got a lovely cartoon representation of lots of different types of viruses and the shapes they might be. And so a virus is made up of genetic material surrounded by a protein shell and or a lipid envelope. So here we've got the genetic material in the center, which is protected by this protein shell, and then a lipid envelope. And we'll learn a little bit more about the lipid envelope in a bit. Viruses require a host organism to reproduce, and they're not considered to be alive. So the way that they do this is they bind to a receptor on the cell. They fuse with the envelope, the membrane of the cell. And from there, they move into the nucleus, and here they can do a range of things depending on the type of virus they are, which I won't get into because that sounds too much like introductory to virology, which is not what we're here for. But in that sense, the viral genome replicates and turns into lots of more viruses. And then it is assembled into a virus with the genetic material and the protein shell. And then as it buds out of the host cell, that's when it gets its lipid envelope, and then it's released. So this idea is that when the virus is infecting the cell, it then creates millions and millions of copies and bursts out of the cell. And so how can we classify viruses? There are lots of different ways to classify viruses. We can do it based on their shape. So we can have this kind of icosahedral shape, an enveloped shape, this more helical shape, or a more complex shape where you've got various different, you've got an icosahedral at the top here and this helical section. We can also do it based on the host range. So some viruses infect just one animal, so maybe they just infect humans, or they infect all animals. We also have plant viruses, and we have viruses that are specific against bacteria. And then lastly, we can classify them based on their genetic material. So some will have RNA as their genetic material, which is one strand, and some will have DNA as their genetic material, which is two strands. And so before we get any further, I want to see what types of viruses people are aware of. And so you can scan the QR code here. So get out your phones, scan the QR code, and I want you to type into the poll there as many viruses that you can think of, and they should appear as a word cloud. If you haven't brought your phone with you, understandably, if you'd like to, you can put up your hand and you can call it out.
I only know one, corona.
Corona, yes. I think that's a very, very common one. So we've got herpes virus, coronavirus, bacteriophage. Nice, someone's been listening. Everyone's heard of the coronavirus? Yes, the 2020 pandemic was the solidification of my future jobs. HIV, Coxsackie virus, nice. Influenza, smallpox, herpes, which is part of the herpes virus, hepatitis, varicella, also known as chickenpox. Nice. Okay. Oh, what's a few more? HPV, lovely. One more person is typing, so I'll let that person get in here before I move on. Oh, okay, they've stopped. Lovely. Okay, so I'm going to run you through. So I thought, and I was right, that the most common one would be coronavirus. We also have common cold, which is actually a collection of hundreds and hundreds of viruses. HIV, measles, influenza, Ebola, rabies. These are all diseases that affect humans. They affect other animals as well, but we kind of know more about them because they affect us, understandably. We have some other viruses that are specific for other animals. So you might have heard of myxomatosis, depending on countries that you've come from, which is an infection that's deadly to rabbits. We have parvovirus, which is a virus that's fatal in dogs, but it's preventable by vaccine. So make sure you vaccinate your dogs against parvovirus. Somebody mentioned smallpox, and also in conjunction with rinderpest. Those are the only two diseases that we have eradicated from the world. Rinderpest is a virus that affects cattle, and we've managed to eradicate it. Then we have tropical viruses like West Nile, dengue, yellow fever, which you might have had to be vaccinated against if you're going to some of those countries it affects. We know that plants can get viruses as well, so we have tobacco mosaic virus and potato virus, which affects the potato. And they're harmful for crop production, which is again why we're concerned about them. Fish can get a type of virus called rhabdovirus. Insects are infected by baculoviruses, and then, as I alluded to, bacteria have their own viruses called bacteriophages. And then we have some other human viruses that you might not have heard as much about because lots of them just live on and in us without causing disease. But sometimes they do. So hepatitis, herpes viruses, papillomavirus, anellovirus, and then, of course, the title of this talk, which is Endogenous Retroviruses. So how can we think about them? We can think about them in terms of how deadly or harmless they are. So you might have heard rabies, which is nearly 100% deadly if you're not vaccinated. Measles is very deadly, hence why we're all hopefully vaccinated against it. Coronavirus and herpes virus, influenza, which kind of affects you differently depending on your age, but also other circumstances. And then some that are harmless, probably you'll only be in bed for a day, like common cold. And then viruses that are harmless or maybe even advantageous, like endogenous retroviruses and the anellovirus. We can also think about what their host is. So these are all mammalian viruses that we talked about. Invertebrates, so dengue, yellow fever, and West Nile, yes, they do infect humans, but their primary host is actually an insect, a mosquito. And the interesting thing about endogenous retroviruses is that they are present in mammals, invertebrates, plants, and other vertebrates like fish. And so that's why they're cool. So the next bit of endogenous retroviruses that I want to talk about is the fact that they're within our DNA. So first off, what is your DNA? And so in 2001, there was a paper published in Nature called Initial Sequencing and Analysis of the Human Genome. And that was essentially a massive project that was proposed in 1999 to sequence the human genome. And up until that point, it was going to be the biggest genome that we had ever sequenced. And it was obviously very cool because it's our genome. And so it was started in 1999. And a draft was published in 2001 where it was about 94% found, like sequenced. And then only in 2016 we were able to have, so the majority of this 94% was done via short read DNA sequencing, which is where we take your big long DNA and we chop it up into tiny bits, and then we try and piece it all together like a massive, really complicated puzzle that all looks the same. And an issue with that is that some bits of the genome are really repetitive, and so when they're all chopped up, you might think, oh, does this repetitive bit here go this space or this space? And so in order to deal with that, we just kind of left gaps. And then long read DNA sequencing was invented, where you can take much longer chunks of DNA. You still can't take the whole chunk of DNA. You can take much longer chunks, and we can read it all the way through. And so this really helped to fill in these gaps that we had here. But the thing is that it took 22 years for it to be completely sequenced, the genome. And by that I mean the 23 chromosomes, the X chromosome, and the Y chromosome. And interestingly, the Y chromosome, which is the smallest one, was also the last one to be sequenced. And this was a collaboration of 20 groups from the US, UK, Japan, France, Germany, and China. And they sequenced, initially sequenced 1 billion and then up to 3 billion base pairs. This sequence data was made available instantly and without restriction, which is honestly why I can do my PhD right now. And I will add in here that the reference genome that we go off, it is a mosaic of a number of anonymous donors, and the majority of those donors were white Europeans because of the biases we have in science and everywhere. And so endogenous retroviruses are a part of your DNA. So this pie graph represents all of your DNA. That's all 46 chromosomes that make up the 3 billion base pairs that exist in every single cell, well, almost every single cell in your body. And so this blue part is going to be annoyingly classified as other, because it generally makes up the structural part of your DNA. Your DNA is really, really long, and we have to shove it into a teeny tiny cell. And so it means that a massive part of our DNA has to be this structural, these bricks, these proteins that are forcing your DNA into teeny tiny coils so it can fit within the nucleus of your cell. And then this red part is called, catchily, non-LTR retrotransposons, or in brackets, jumping genes. Now, jumping genes are really cool. They were discovered by a woman, which is awesome, and that's why I put a kangaroo there. They have been largely thought also to be a big part of junk DNA. We now know that isn't the case, but I'm not going to be talking about these specifically today, but they're still interesting. Now, you might be thinking, Erin, where's the part that makes up me? What part did I get from my parents? Well, you got all of this from your parents. But the protein coding genes, the bits that make up the cellular machinery of you, so what makes your hair color the color it is, what makes your eye color, what provides the genes to make sure that you can digest lactose or not, is encoded by just 2% of your genome. And that is what makes you. So it's a surprisingly small amount. And when we sequenced it in 2001, we were very shocked to see how small it actually was. And then the bit that we're interested in today is endogenous retroviruses, which make up 8% of your genome. They are the remnants of viruses, and we are very interested in how they affect us in health and disease. So a little bit of etymology for the literary students in the room. So endogenous retrovirus, we can split it into endo, which means within, as in within us, as opposed to exo, which is outside of us. Genus, which means arising from, and then retroviruses, which is a family of viruses. The most common one you've probably heard of is HIV. And we'll focus on the retrovirus part of it first. So this is what I showed at closer to the start, and don't get too bogged down in the details, but essentially it's this idea of the virus coming in, binding and fusing with the cell membrane, and then they're called retro because they go back. So they have RNA that they transcribe back into DNA. Our cells can only go DNA to RNA, but they have this enzyme called reverse transcriptase, which means they're kind of special, and they can go from RNA to DNA. Now this DNA then enters the host nucleus and actually integrates into the host genome, which is why HIV can be so devastating, because the DNA of the HIV virus is integrating into your DNA and causing havoc. And then here it does what the other viruses does, which it makes more of itself. They then assemble, they bud, and they release. So that is essentially what makes a retrovirus. And the most important part is this integration here, because without it, it wouldn't be in our DNA. So this little bit here is just the previous slide. Don't worry about it too much. But basically, ancient retroviruses infected and integrated into our germline cells. And the reason that's important is because if it just infected your lung cells or your heart cells or your brain cells, it would die with you. Unless you passed it on to another person and it affected their brain cells, their lung cells, their heart cells. That's the only way that virus would transmit. But this one goes into the germline cells, so the eggs and the sperm, and that means it is passed on to the next generation. And so I'm showing here ancient primates, which it did this in, and then this integrated, we now call it a provirus, but it's essentially a viral sequence, is passed from generation to generation. And then as we get multiple generations, we get more and more of these infection events, and our genome eventually becomes filled with viral sequences. There's all these colors here. All right, so when we first found these, we did, I think, like all people go and just go, well, I don't know what that is, so I'm just going to shove it away in the closet and not think about it. So there was a big thought for a while that they were just junk DNA. However, we now know that endogenous retroviruses are a vital part of our genome in many ways. So we know that they can be involved in turning genes on and off. We know that they interact with other viruses. We know they interact with human cells. And then this results in a variety of processes, such as driving evolution, having resistance to viral infections, and also the development of disease. And I'm going to go through some sort of like examples of this so you can properly kind of visualize and relate to your own life, hopefully. So number one, the evolution of the placenta. We wouldn't be here, well, we definitely wouldn't be here without retroviruses, but this is one of the reasons why, and it's very cool. So we're going to have a little quick sex ed lesson. So egg meets sperm, fertilizes, divides, so turns into 2 cells, which turns into 4 cells, which then eventually turns into blastocyst, which embeds itself into the uterine lining of the uterus. And it is this link between baby and the placenta. So the placenta is developed by the mother. It's cool. It's an organ that is created for this process. And an issue is that this baby is made up of 50% foreign DNA from the father. And in any other circumstance, your body would go, no, get rid of it. If you've heard of people getting organ transplants and they need to take rejection drugs, anti-rejection drugs, because of that process. Obviously, pregnant women are not taking anti-rejection drugs, so how is this working? And so it's all responsible due to a gene called syncytin, which is difficult to say. But essentially it's a gene that is derived from the envelope, so this lipid coating, which allows it to fuse with the cell, and from an endogenous retrovirus. And like I was saying, the viral envelope of a virus is made up of lipids that mediate this fusion between the virus and the host cell, which is also surrounded by lipids. And in doing so also dampens the immune response, because if you're a virus, you don't want to alert the host cell that you've entered. You wanted to do it on the down low. And so syncytin has been co-opted during human and other placental mammal development and evolution to allow this placental layer to fuse with the uterus without activating the immune system. Because if you can do it on the down low, then you'll be allowed to grow. And there are other genes obviously involved in this whole process, but this is a very key one. Okay, so if you eat a piece of bread and you put it on your tongue and you don't like chew it up or swallow it, it will slowly dissolve into sugar on your tongue. And that is because we have amylase, which is an enzyme that digests sugar, in our salivary glands. However, it wasn't there originally. So we originally had this amylase gene, lovely structure here. It used to just be in our pancreas because that's kind of where it wanted. You eat it, it goes into your stomach, the pancreas secretes enzymes that digest it, and that sugar goes into your bloodstream and helps all your cells and everything. But there's an endogenous retrovirus with the catchy name of A1C, which it acts as a different promoter for this enzyme. So all genes in your body, they get turned on and off by promoters, which are essentially just that alarm clock that gets them going. And this ERV-A1C managed to, it got inserted in a place where it meant that amylase was secreted in your salivary glands. And this also coincided with the times that humans, after the agricultural revolution. So the idea is that this happened to one human or a few humans, it was advantageous, and then those humans survived, and other humans who didn't have this gene did not survive. And it allows animals and humans to digest starch more efficiently, which starch is the reason is we need energy, so it makes sense to be able to digest it efficiently. Okay, so if you have heard of stem cells, which are, I think if you, they're a very important part of the developing person, and you as an adult, but also in terms of treatments for various diseases. Now, the idea of a stem cell, and specifically embryonic stem cells, is that they have the capacity to differentiate or change into any cell in the body. This is very important in the embryo, where they start off from 1 cell, 2 cell, 4 cell. You need a way to make sure that those cells can become the brain or the liver or the foot. And we have been able to extract these human embryonic stem cells from blastocysts. And they found a really cool thing, which was that another catchy name of H expression in these embryonic stem cells was much higher than other ERVs, so ERV-K here, which is just another example. And it's not seen in other cell types. So HeLa cells is a cancer cell line that is used for many, many things. And K562 is also another cell line, but they're not embryonic cell lines. HESC, or human embryonic stem cells, are the only one. And so obviously ERV-H is massively upregulated. Yes. And so on that, to look at it in a little bit more depth, what we can do in science is, if we're interested in what the role of a gene does, is we like to delete it and then see what happens. And so what they did is they took some normal embryonic stem cells and then they silenced the ERVs. And you can see that they look different. And the way that they look different is that we know that these ones look like normal embryonic stem cells. And these ones look more like fibroblasts, which is a differentiated cell type. All us adults here in our connective tissues have fibroblasts. And then the next question was, well, okay, what is ERV-H doing? And they eventually found that ERV-H was being expressed as a very long piece of RNA, and it was linking these two very important proteins in making something a stem cell. And what that means is it was making sure that this connection between the two proteins happened, and that we could have these expression of these lovely sets of genes with great names, and that makes a cell a stem cell. And so when we take that away, the P300 and the OCT4 can't join, and we don't get this expression that we see. Okay. I said that it protects us from viral infection. And one of the most interesting, I think, is protection from HIV. So both HIV and these endogenous retroviruses are both retroviruses. So there's much more similarity between them than, say, coronavirus or flu. And what they took is they looked at the expression of this ERV-K102. And I'm throwing a lot of like ERV numbers and letters at you. They don't mean anything really. They're just different. Think of them as one's like influenza and one's like HIV. They're relatively different. They took people who were HIV positive, either on therapy or not on therapy. And then they took people who they considered to be immune to HIV, because they were commercial sex workers who had been exposed to HIV during the course of their jobs for at least 3 years. And they found that they did not have HIV as they might have expected. And in these individuals, compared to those with HIV, the ones that were protected from HIV are in pink. And you can see that they've got much higher levels of this ERV-K102. So an average of 4.21, sort of relative expression, it's not an absolute number, compared to around 1. And so obviously, as scientists want to do, they went, why? And it could be that, we don't actually know, but there are theories. It could be that this ERV-K is so similar to HIV that it blocks these receptors, and so HIV can't actually enter the cells. It could be that when we assemble this virus, that the ERV-K envelope protein likes to come and swallow up the HIV DNA, and that's not going to be an effective HIV virion. And then another one is that somehow ERV-K is interfering with this budding and release of HIV. But it's hard to study. And finally, and we're getting more into my work here, is the contribution of endogenous retroviruses to the development of autoimmune diseases. So autoimmune diseases occur when the immune system starts to attack and damage your own healthy cells. And there's over 80 different types, and they affect different regions in your body. and symptoms are very variable, and also our understanding and thus our ability to treat these autoimmune diseases is quite limited. And one of the ones that I'm particularly interested in is multiple sclerosis. Now, multiple sclerosis is a chronic autoimmune demyelinating disease, and I'll take you through what each of those mean. But chronic means it's a lifelong disability, and diagnosis occurs generally between the ages of 20 and 50. Autoimmune, like I said before, means that the immune system is attacking normal tissue. And then demyelinating means that the immune system is attacking the insulating layer that surrounds brain cells, and it means it is a disease of the central nervous system, or the brain and the spinal cord. And so essentially we have these immune cells that are being transported into the brain. There they cause damage to these neurons, and specifically this myelin sheath, which is on the outside, starts to get damaged. And then this is an example of an MRI that we see in people who are living with MS. And you might not have seen an MRI before, but these white patches should not be there. Your brain should be a nice healthy grey colour on MRI. And again, why I'm interested in it is because Canada has one of the highest rates of multiple sclerosis in the world. And this is partially to do with a latitude gradient. So the darker the colour, the higher the rate of MS. So you can see in the US and Canada here, it's red. Up in some Nordic countries, it's in a dark red. And then as you get closer to the equator, you have decreasing prevalence of MS. And this is related to exposure to vitamin D. So in Canada, for the majority of the year, we do not receive sufficient vitamin D from the sun because we're so far north. So make sure you take your vitamin D supplements. And so there are other risk factors that contribute to the development of MS that take place across your life. So we have stress, we have infection with viruses such as EBV or Epstein-Barr virus, which was the topic of my talk last year. Changes in the microbiome, which we've already alluded to. Passive smoking, vitamin D, which we talked about, and then this all works in conjunction with your DNA. And so we are continually observing a correlation between another ERV expression and MS. And I'm not saying to look too deep into these graphs, but it's essentially to say that it was first observed in 2012. We have healthy individuals here without MS, and then individuals with MS, and then relative expression of ERV. And we can consistently see that MS cases are higher than healthy controls. And so we saw that in 2012, 2020, and there are obviously a whole heap of other studies in between. And then just recently, this year, we saw that we see higher, when you have increasing disability with MS, that's when we see the higher ERV expression. But unfortunately, with this correlation, we still don't know why. And this is kind of an overview. I'm not suggesting you look too far into it, but it's just to say that we have some ideas, but we don't really know, and that's why I'm doing a PhD on it. We know that there is some interaction with herpes viruses, specifically Epstein-Barr virus and ERVs. We know the different cells that might be involved. We know that the ERV might be secreting a protein, which could be neurotoxic, so it's affecting the microglial cells in the brain. We know that T cells are also involved, and they like to cause a lot of damage wherever they go. And we also think that there is this interaction with the protection that we have between our brain and the rest of our body, because the brain is very protected from everything. That's why when you get a brain infection, it's so devastating. This is just another view of that to show how perhaps the ERV protein, envelope protein, is causing this damage. And so what can we conclude? We can conclude that endogenous retroviruses are a large and essential part of our genome, and that they were vital in our evolution and remain an important part of having a healthy body. But we also know that when they become dysregulated, we see the development of various diseases, such as multiple sclerosis. And we still don't know much about our virome and how it affects us during health and disease, hence why we have lots of lovely people studying it. And with that, I'd like to thank you for listening, and thank you to my labs. We went for a little, we hugged some puppies, and I will take any questions you might have.